So as Greg said, I'll be talking about some technology that's still under development. So this is a session looking at things that are, I guess, have a horizon of 3 to 5 years from now. And specifically, I'll be talking about what we're, I guess, terming the tricorder project. But it's also trying to address some things that I think are fairly common questions that all of us have when we're shopping, when we're eating, or when we're talking over food. And this has to do with a lot of the things that are important with respect to food. What is it that gives food its color or flavor or aroma or taste? Many of those things are not listed on food labels. Many of us don't know what those compounds are, and they're obviously very key to consumer decisions, but also with respect to the health effects or purported health effects for foods. Similar issues with respect to the things that the micronutrients that Greg mentioned. Many of them are not labeled. Information isn't available on those, in part because it's very hard to measure them. Same issue with contaminants, food adulteration, authenticity. These are challenges that require a lot of skill and a lot of effort to be able to measure or test. And the same thing with respect to pesticide, herbicide residues, and other contaminants that have actually already been mentioned today. How do we know what's there? So these are things that are relevant to food safety, to food quality, to food and health. And questions that are, as I say, asked by many of us and by many agencies. Greg mentioned the Food Metabolome Project. This is a recently funded effort coming through Alberta Innovates Biosolutions and Health Solutions, also supported by ALMA. And it's, I guess to put it simply, the metabolome is the parallel for the genome. And when we think about the genome, that's the collection of genes that we find in organisms. The metabolome is the collection of metabolites, or small molecule chemicals that we find in organisms. And the organisms can include, apples and bananas, or meat, or living systems, such as ourselves. And so if we just take the word metabolite and drop the ITE and add an OME, now we've got an omics platform. And so that's what metabolomics is about. And it's something that we've been doing in Alberta for a number of years. And believe it or not, in fact, Alberta is considered a world leader in this area. What we're trying to do is determine the compounds that we can find, both in terms of the amount and the type, in the various foods that are produced in Alberta. But we're also cataloging this information for many other foods from around the world. So in metabolomics, there are many applications. Typically, if we were doing some analysis of food, we would take the food products, tissues, or samples. Metabolomics can also be applied to human health and to animal health as well. Those samples are extracted. We get what we call biofluids or extracts, and then they're sent in for chemical analysis using a variety of platforms that we maintain at the University of Alberta. And then these spectral collections are analyzed and interpreted. And it's a fairly difficult process, fairly time-consuming. The technology that we typically use involves mass spectrometry, high performance or high pressure liquid chromatography, NMR spectroscopy, liquid chromatography mass spectroscopy. And with these techniques and with software that we've developed and others have developed, it's possible to measure more than just a couple compounds. In fact, we're measuring hundreds and sometimes even thousands of compounds from a single sample. And this slide just illustrates the types of technologies, the sensitivity going from millimolar, micromolar, nanomolar, picomolar levels, and the numbers of compounds ranging from hundreds to thousands to tens of thousands of compounds, depending on the technique. So this is a fairly new area. It's one that has a lot of people excited, not only in the world of agriculture and food, but also in health, medicine, and veterinary applications. So in terms of the work we've been doing on the food metabolome, measuring the compounds that make up the food, well, you can tell your friends that there's about 30,000 different chemicals or compounds in the foods that we eat. No single food has all of them. On average, there might be 3,000 different compounds in a given food component. We've cataloged those. We've got structures for them. We've got descriptions for them. We've got alternate names for them. We've got information about their food, flavor, aroma associations. And we've got content information for about 3,500 different foods. We're also compiling data on food and health effects and entering that data into a publicly accessible database, which will go online or live probably in fall, although there are examples that we have right now. We just want to make sure the data is completely correct. Now, when we're doing food analysis and following that metabolomic workflow, one of the things that's very obvious is that the instrumentation is big, expensive, clumsy, and requires a lot of skill. The average instrument weighs half a ton. Some of the instruments cost more than $1 million. To operate the instruments, you need a PhD. To repair them, you need a PhD. As a result, a lot of the analysis, the work that we're doing is expensive, and the work that's being done elsewhere is also expensive. The instruments aren't portable, and it's difficult to do. So the goal, and this is where the nanotechnology comes in, is to try and develop systems that would be portable, something that could be run in the field or in a small office or in a small company where the instruments are cheap, the costs for tests are cheap, and that anyone could use them. So what we're wanting to do, and through my associations with the NRC and National Institute for Nanotechnology, we're starting to work with a consortium of scientists in NINT and the University of Alberta to exploit some of the technologies that have been developed here, things like lab-on-a-chip technologies, what are things called nano-electrical mechanical devices, or NEM systems, microfluidics, biosensors, and then to try and integrate some of these things to perform the processes that we do in my lab, which involve extraction, separation, and detection with these $1 million instruments. So putting all of that together and making that something small enough creates something that I think all of us have sort of seen at one time or another, the tricorder. And whether it's something that Spock is holding down at the bottom or something that's perhaps a little more modern looking like the iPhone, that's what we'd ultimately like to generate. So we're exploiting things that in some cases already exist. There are things like capillary electrophoresis, or CE on a chip, high performance liquid chromatography on a chip, gas chromatography on a chip. These are things that you can buy. These are things that can also be made with the tools that we already have in NINT and at the NRC. But we also have to have things that can detect those things. These things are great for separating. We want to be able to measure those separated compounds. And so a number of people in our consortium working at NINT, like John Davis, Stéphane Avoy, Ray DeCorby, are developing a variety of tools that are like spectrometers, mass spectrometers, things that wiggle or vibrate when something small, like a molecule, binds to them. And when they are bound to a small molecule, they wiggle or vibrate at a different frequency. And so this way we can actually make mass spectrometers that fit on the head of a pin, basically. So these are the tools that we're trying to integrate and have actually had some interesting success. So that's one aspect of the development. And this sort of highlights how these things might work and do work. Typically these little cantilevers or resonators are decorated with a variety of surfaces that bind to the molecules of interest. They could be antibodies that bind to small molecules. They could be other types of proteins. They could be other polymers that are selective. When they bind, the resonators, as I say, shift their frequency. We measure the frequency. That interprets the mass. That allows us to identify them. So it's a simple concept, but it's reducing the size of the instrument to the size of a refrigerator to something that fits on the head of a pin. Another approach we're using is exploiting things that nature developed. Many small animals, including microbes, have proteins that bring in specific compounds into their systems, and bacteria bring in lots of nutrients. And there are proteins called paraplasmic binding proteins that bind to a number of the small molecules that we're interested in measuring. So we want to clone those proteins, optimize them, and make them into essentially biosensors that we could attach to these cantilevers or that we could put into simply solution phase systems. One of the things we can do is exploit fluorescence, natural fluorescence, using a variety of green fluorescent proteins, and these can be mutated to change to anything from cyan to emerald green to yellow to orange to deep red. In essence, these proteins that are shown here can be adjusted, and combining them with the paraplasmic proteins, whoops, I guess we lost an image here. Not sure what happened there. But combining them with these paraplasmic proteins, we can change the fluorescence. And so we can get different colors for different metabolites. And so, again, you can go from $1 million room-size instrument to something that's as small as a little Eppendorf tube to measure it. So the principle, as I say, is to try and reduce the size, the cost, the efficiency, and the simplicity, make it simpler, to measure these compounds. And as I say, the applications can be in food and food testing. They can also be in animal health and also in human health. So the timeline here is already we're working on things like what we call the fluorescence-based biosensors. Those little oscillators are also being developed, and then working on some of the chip-based separation and extraction. So these three components are going to be brought together beginning 2014 in terms of the device integration, and then to hopefully start to take that out to the real-world testing by 2016. And so with that, we'd hope to have something that could be used, as I say, in the field or in the office to help with food quality, food safety, and general food chemical assessment. So with that, I'd like to thank the people. There's a lot of people involved in the tricorder development, about 50 in all, and some of the funding agencies that are supporting the project on the food metabolome and other aspects of food and health measurement. And thank you for your attention.
Are there any questions for Dr. Wishick?
And what's the timeline do you see getting something that might be first introduction to a company that might be able to take it out as a product?
Probably what we're trying to do, I guess, is what's not shown on this timeline, is to make things largely sellable as a kit initially, so that the kit would have some of the reagents that then either could be analyzed on some of the more expensive instruments, but the kit is cheap. And so either they rent time on the instrument, then run the kit. So that way, at least it segues into the idea of inexpensive testing. The device still needs to go through, I think, some field testing. So I wouldn't imagine that we'd have the prototype ready until about 2014. And I think that's at the stage when we'd like to say, okay, come on in, take a look at this. Does it look interesting to you? But it's sort of a stage progress, which is to release the kit, which is about a year away, and then to follow on with devices that would be portable.
Yes?
Dr. Wishart, how do you see the potential, our ability to analyze has now exceeded our ability to interpret. So at what stage do you see maybe this going into personalized medicine, where you'd say we can test each food item for 3,000 metabolites, but only one or two are important for your genetic makeup or for this specific gene interaction? So how can you convince, do you see a whole new area of science or a whole new career of people who have to help translate what we gain from our knowledge into real application?
I guess I can see that emerging. I mean, this is something that's happening obviously already in medicine with personalized medicine and some of the genetic testing that goes on, and we need genetic consultants. I can see this actually becoming more integrated into essentially nutrition and the training that nutritionists have, but also the idea of this food database or food DB is to make that information very public and very accessible. So the database itself has some angles that probably would be more interest to scientists, but we're associating the database with a lot of really simple, to the point, what we call food fact sheets, to try and get that information to people. Whether it now goes to the next stage, we're associating what you should eat and the doses of dark chocolate or blueberries or whatever that might be healthy for you. That could come out of this sort of work. But that's still a ways away. Clearly, there are people that benefit a tremendous amount from certain types of foods, and this is something that's becoming more and more evident as we do more and more work on it. And a lot of this is being done in Europe. and the information is sort of trickling to Canada and North America. But why does the Mediterranean diet allow people to live that much longer? And it's specific for certain types of people, so it seems to be this interplay between your genes and the food that you're eating. And we'd like to know exactly what is best for certain people. So, good question.